Welcome back to CAPSA, your trusted channel for neuroscience and pharmacology lectures. In the previous video, we introduced the vesicular monoamine transporters VMAT1 and VMAT2, their type, structure, and tissue distribution. So today we move on another step and dive into the mechanism action of VMAT. In this lecture, we will explore how VMAT transports monoamines into vesicles, the role of the proton gradient and ion exchange mechanism, the step-by-step transport cycle of VMAT, and finally how VMAT actively or VMAT's activity is regulated under the physiological and pharmacological conditions. So let's decode the mechanism that controls the neurotransmitter storage at the molecular level. To understand VMAT function, let's first talk about energy source that powers its transport. VMAT does not directly use ATP. Instead, it is a secondary active transporter, meaning it depends on gradient created by another energy dependent pump. Here, that is the vacuolar type, so hydrogen plus ATPase or V-ATPase, which uses the ATP to pump protons into the synaptic vesicles and creating an acidic vesicular environment with a pH of around 5.5. This steep proton gradient is then used by VMAT. As protons exit the vesicle via VMAT, monoamines from the cytosol are pulled inward. This H plus out and neurotransmitter in movement defines the antiport mechanism of the VMAT. Now let's talk about the transport cycle of VMAT in sequential steps. So the first V-ATPase acidifies the vesicle by pumping in protons. This establishes a high internal H plus ion concentration. Then a monoamine molecule in the cytosol, like the dopamine or the serotonin binds to the outward facing side of the VMAT. This binding triggers a conformational shift in the VMAT protein. In this new conformational shift, protons exit the vesicle through the VMAT, moving down their concentration gradient. Simultaneously, the monoamine is translocated into the vesicular lumen. After release, the transporters reset to its original configuration, ready for another cycle. This cycle is rapid and continuous and allowing neurons to efficiently fill vesicles with neurotransmitters in the preparation for the synaptic release. A very important point in VMAT function is the stoichiometry of the ion exchange. For each monoamine transported into the vesicle, VMAT exchanges two protons out. So the ratio is 2H plus per one monoamine molecule. This makes the process electrogenic, meaning that net charges are moved across the membrane, contributing to vesicular membrane potential. Note again, VMAT itself does not consume ATP, it relies entirely on the electrochemical gradient established by the V-ATPase. VMAT activity is tightly regulated to meet the neuronal demand for the neurotransmitter storage. One major factor is vesicular pH. If the interior pH become or interior acidic concentration become less acidic, the proton gradient weakens and VMAT efficiency drops. So the membrane potential of the vesicle also contributes to the driving force. Additionally, post-translational modifications such as phosphorylation can regulate the transporter's activity and glycosylation can affect its stability or trafficking. Moreover, during high frequency synaptic activity, feedback mechanisms can modulate transporter function to balance the storage and release. To recap, VMAT is a secondary active transporter using a 2:1 antiport mechanism powered by the vesicular proton gradient. It plays a crucial role in monoamine storage and enabling neurotransmitter release during the neuronal signaling. This transport is regulated by vesicle acidity, membrane potential, and various protein modifications, ensuring that synaptic communications remains finely tuned. If you are finding this series helpful, be sure to subscribe to channel CAPSA and to get notified about next upload. In the next video, we will dive into the clinical relevance and pharmacology of VMAT, exploring how drugs like reserpine, tetrabenazine, and others drugs impact on monoamine levels in the brain. Drop your questions in the comments. Thanks for learning with CAPSA. See you in the next class.